So, are you ready for mods? A whole year of studying, and now it's time for our first proper exams.
I am panicking. I have not done enough revision. I don't know the difference between Sarcodina and Mastigophora, and have no idea about the role of glycosylphosphatidylinosyl in anchoring membrane proteins. Oh dear.
But surely you must remember some stuff. I mean, you were always wide awake in lectures, weren't you?
Well, I get easily distracted. My mind always wanders to thinking about signaling transduction pathways, even during all the Wirtz lectures. Receptor tyrosine kinases are just so goddamn sexy. Just thinking about ligands binding to their extracellular domains causing dimerization and trans-autophosphorylation of tyrosine residues on their cytosolic tails makes me all hot and sweaty. When Graham Taylor is going on about tetrapod evolution, all I can think about are those sweet little adapter proteins binding to activated receptor tyrosine kinases by their SH2 domains. And those adapter proteins, such awesome intracellular signaling proteins, also have SH3 domains which allow them to interact with other proteins such as that hottie, the guanine exchange factor. And those GEF proteins activate GTPases.
Wow. You are very odd, aren't you? And signal transduction is far too complex and confusing. I mean, why does the RSGTP activating protein actually deactivate the RSGTPase? It makes no sense. Anyway, this is enough about your weird obsession with signaling pathways. How is your organism's revision going? That is our first exam. Surely you know most about that?
No.
But you must know the differences between the earliest synapsids, the pelycosaurs and therapsids, and the Mesozoic mammals.
No. Do tell me.
Well, the Mesozoic mammals were all quite tiny, pathetic little things. Although, unlike the pelycosaurs and therapsids, they have true mammalian characteristics such as fear, lactation, and suckling. They also had a Hadrian gland. In mammals, this secretes lipids which are spread across the fur during grooming and provides a hydrophobic insulatory layer.
Apart from the evolution of mammalian characteristics, are there any general trends that can be seen during synapsid evolution?
Oh, yes. The temporal furniture get bigger. There is greater teeth specialization. A secondary bony palate develops. This separates the respiratory passage from the mouth, allowing for simultaneous eating and breathing. There are also changes in thermoregulation. Early synapsids did not regulate their internal temperature as well as modern mammals. The pelycosaur was in fact an ectotherm. Many had large sails on their back, formed from an elongation of neural spines. This structure had a role in temperature regulation.
Well, it has been very nice talking to you, but I really must go and do some revision. I will watch Snakes on a Plane to help me remember what squamata are. And then maybe the human centipede. Surely that will have lots of useful invert information that I can include in my mods essays.